So hello and welcome to second session of fundamentals in bioinformatics, getting started with bioinformatics in R and in Python. So today we are going to review the fundamentals of DNA code, what is all about DNA and what do we know about it and what kind of answers we can find from there, and in generally how to play around with the DNA code in a programming environment like R and in Python. So let's review what do we already know about DNA code. So just like a book, the DNA code is organized as letters that form sentences and sentences that form paragraphs and paragraphs that are organized into chapters. So knowing how to read this code and identifying patterns with this code will help us decipher the full story about generally who we are or who the organism is, and look into our history and look into the variants and look into variations in certain regions of DNA that will define the characteristics of our phenotype or some disease stages. So understanding how we are by understanding the DNA code becomes extremely important to know how we are connected to other living things around us. So DNA, as you all know, is short for deoxyribonucleic acid. So it is a long molecule composed of two chains that coil around each other to form a double helix, carrying the necessary genetic instruction for the development, function, growth, reproduction, and all the other functions of all known life. This includes both multi and single cell organisms, as well as many bacteria and even some viruses also have a DNA code in them. So for example, in human DNA, we almost have 22,000 proteins that are encoded in 3.2 billion letters in total in one strand of DNA. And yeah, so even more, if you break the DNA molecule into its molecular basics, fundamental level, we know that DNA is made up of nucleotides. So a nucleotide is made up of three components. So there is a sugar component, which is called deoxyribose, and there is a nitrogen containing base, which we already know and which we name the DNA base with. They are adenine, thymine, guanine, and cytosine. And then these are all attached to a phosphate group. So the organization of nucleotide base is such that the nucleotides are joined to one another in a chain by covalent bonds that form between the sugar of one nucleotide and the phosphate of the next. This results in an alternating sugar-phosphate backbone. So the nucleotide of two complementary strands are connected together by base pairing. So this base pairing scheme, which always connects adenine with thymine and guanine with cytosine. So when referring to number of base pairs contained in a sequence of DNA, we are actually referring to these connected bases. So each strand of DNA molecule contains a sequence of bases that is complementary to the bases in the opposite strand. The pairing, this specific pairing between adenine and thymine and guanine and cytosine is the reason DNA gets its characteristics double helix shape. And this complementarity also gives rise to the possibility of copying over the code several times without any percentage or without very low percentage of error. And always the forward strand or DNA strand is read in such a way that we read from the 5' end of the nucleotide chain to the 3' end in the forward strand, and in the reverse strand it goes upside down, 5' end to the 3' end in the reverse direction. So from this DNA molecule, which is absolutely common to all different kinds of cells and all different types of cells, we get different types of cells and different cells that are phenotypically different and that are functionally different. So all the necessary codes for them to be functionally different and phenotypically different are contained in the DNA. And the way in which the molecule presents itself to the to the transcription and translation of converting these DNA into protein decides which cell will form which function and which cell will take up which phenotypical characteristics. So this is described in a central dogma of molecular biology, which includes a two-step process, transcription and translation. So by which the information from the gene or from the DNA flows into protein via an intermediate step of creating RNA molecules. So in each step of this transformation, we see how the DNA code is generally used. First, the source code or the source DNA code or source DNA molecule is transcribed into a working repository of RNA where some of this code is modified. And then this working repository of mature RNA are released into the cytosol, released from the nucleus into the cytosol for the production of protein, which takes place using translation process. So each gene in general is a recipe for a certain protein so that it encodes the instruction from which a protein can be made and how different these proteins can be. And even within the same protein, there can be multiple forms or multiple isoforms of protein which are necessary for different at different time periods of the cell's differentiation and cell's functionality. So these codes of DNA, or how the DNA code encodes for different amino acids that forms a protein, is through a sequence of trinucleotide codons. So these trinucleotide codons will include will correspond to different amino acids at different level as at different combinations as mentioned in this table, and also includes a stop signal which will signal or which will interrupt the production or the synthesis of the protein in the cytosol. So once the right pieces of DNA is copied over to messenger RNA and is prepared for translation, then the messenger RNA is released for that specific protein, is released to the cytosol, and then this messenger RNA, which contains these codons that are translated into amino acid blocks one by one, will then be responsible for the formation of the protein that is synthesized in the cytosol. This process this process takes place with the help of RNA polymerase and the other necessary enzymes in the RNA polymerase complex. So to get a general idea about the genome composition and the gene sizes, so we have a variety of different organisms with variety of different sizes of genes, and that does not directly translate into the complexity of the organism at all. For example, a simple influenza virus may contain only 11 genes, but human as will contain 22,000, anywhere around 22,000 proteins or genes, and a grape sequence may contain 30,000 genes. So in addition to the number of genes that increases the complexity of different organisms, there can also be different sizes of these genes within the same genome. So some of the protein may be really long as 2.3 billion or 2.3 million nucleotides long in CN, TN, AP2 gene, and some protein may be as small as 500 nucleotides long. So these complexity are the main reason for the diversity and for the different organizational complexity and variety of different organisms. So in general, if you take a single gene structure of a eukaryotic organism, it might comprise of upstream and downstream regulatory sequences that will control transcription, and even inside the gene sequence itself, there will be codons or there will be introns that will undergo splicing before a gene is translated into protein. the extraordinary length of some of the genes will enable the presence of introns in such a way that it gives rise to a mechanism of alternative splicing, which can give rise to different forms of protein or different isoforms of protein from the same sequence of from the same sequence of the gene. So these alternative combinations of exons and different lengths of the isoforms of the protein that gives rise to the that is resulting as a result of alternative splicing also increases the complexity of number of proteins that can be generated from a gene structure. But this is not the case always. But so far, the biology of the gene and biology of how the genes are code for protein has been very well studied and is still being studied. And we also know there are different ways to study this. But how does studying DNA sequence in a computer coded environment will help us understand the sequence itself and help us understand the organization of the sequence itself is left to be explored a lot. And to start in that way, we have to convert these biological samples into digital data. So one way in which these biological samples are converted into digital data are through high-throughput sequencing technology or next-generation sequencing methods, which take these biological samples and then they process them through sequencer machines that will call for the DNA bases at each and every position, so that these calls are recorded in a text-based file format like FASTQ or FASTA and GTF. There are several file formats that is associated with the building up of a genome sequence. So the sequencer will give its output as FASTQ file formats of short fragments of reads, and once these short fragments of reads are then subjected into a sequence of analysis known as pipeline analysis for genomic data, and then we can build a whole genome sequence of any organism of any targeted loci in a genome. So this whole genome sequence of the organism can be stored in a format called FASTA file format, which contains or which starts with a greater than symbol as the first at the first line that mentions or that indicates the first line is a header which can contain or header that contains necessary information that describes the sequence itself. The information can include accession ID of where to find this specific sequence in a related database. It can include, for example, the genus name, the species name of the organism. It can also include the method with which this sequence is built. And it can also include, for example, whether this piece of genome is a whole complete genome, or is it a piece of or is from any specific gene, or is from any single chromosome, etc., etc. And then what follows in the from the second line is the actual sequence itself in the order in which they are called by the sequencer machines, or in the order in which they are mapped and assembled in the assembly pipeline. So this is the file, this is the DNA sequence input that we will be concerned with while we are working in a programming environment. So let's stop the introduction to DNA sequences at this point. Then I will now proceed on to onto the associated lessons, which will explain or which will give you a hint to how to work with the DNA sequences in a coding environment like R.
That's it. Shall I stop?
No, this is only for the first part. So I have to share everything for them to know about this. That's why I have stopped here.
Okay.
1 second. Okay.
Yes, sir.
I have to open a couple of tabs here so that I can show them where to find what. That's all. Okay. Can you see my screen? Can you confirm what do you see?
Yes. Yes, sir.
You see one screen or two screen? Two screens?
So I can see the portal, the program page for our.
Only that one, right?
Yes, sir.
Okay, perfect. Thank you. So let's start again. So welcome back everybody to the second part of today's session, which is a hands-on session of reading DNA sequences in R and manipulating the sequences to understand the variety and understand the diversity in these reading sequences. So the associated lessons can be accessed from these links, and now we are going to access a last part of the lesson that we were discussing in the previous session. In the previous session, if you remember, I stopped at understanding, or I stopped at loading data into R and accessing different parts of the data frame or different parts of the data using numerical indices. Now, this associated resource also has a an introduction to function which actually tells you how to work with DNA sequences in general. So I explained to you in the previous class or in the previous lesson that the object that we read into R can be a numerical object, it can be characters, or it can be string object, or it can be a collection of numerical objects, that is array. I think it's here, maybe, yeah. Okay, it's not here, sorry. So collection of numerical objects or columns, that is array or matrices, and it can also have a collection of categorical and numerical and string and many different object class types that can be collected or that can be loaded into a data frame, which we did in the last session. So now and let's see how we can work with functions in R. In generally, functions are used for that specific piece of code which we tend to use multiple times. For example, let's go to my R code. So in your R Studio, as I had explained to you in the previous session, the organization could be a little bit different. I have the console window up here and the source window down here. So yesterday I explained to you the 4 different panes of R Studio, where this is a console window which we use to interact with the R console. Let's zoom in. Yes, I think yes. And this is the source window where we paste our sequences so that we can execute them later. So for example, if I am concerned with calculating or counting the number of characters in each and every one of these sequences, then I can use 2 or 3 different ways in which I can calculate these sequences. For example, let's load up this specific DNA sequence into an object. Let's clear everything. So yeah, that object can be seq1, and seq1 is being to be loaded with this, and see, well, this one work. Yes, the seq1 will work as this. So if you print seq1 is going to print the contents of the sequence as this. So now you can actually take the or count the number of letters in the sequence using nchar object seq1. So nchar should actually give you number of characters or number of letters in in sequence 1 or in any sequence. But we can also use, or we can also encapsulate this nchar and other ones into a function, so that we can use it in a better way. This is not a great example of using a function, because whatever this function will do can be performed using this inbuilt nchar function in themselves. So, but while we go through the other resources, then other codes, then you will have more introduction or more information about how this function is going to be used and why we need function. If you have to, for example, if you have to calculate distances between 2 points in any DNA sequence, then you can build a function for that, and you don't have to. That function can have as simple as 3 or 4 lines of code, and you can repeat or call that function multiple times as to use it better. So how to do that? So functions are generally defined as the function's name here, and then followed by whether or not it is a function in R. Can you see my screen? Yes, in R, exactly. So this argument function, or this specification function, tells the R console that we are going to define a function, and whatever is inside the bracket tells R that we are going to pass something to the function. So this object that is going to be passed into the function can be anything, and here we are going to define that as seek. And inside the function, we are going to define what we are going to do with the argument object that is passed into the function. So in this case, we are simply calculating the number of characters for this specific sequence object, and then storing that number of characters into an object by the name length. And then we are printing the object length to so that we can get the result of this number of character of this specific object sequence. So first we have to run this. In order to run this, we can block all of this and then press the run selection or run current line option. So when you do this, you will not get any output or any reply from R. But if you check in your environment pane, you will see that there is a function that we have defined, and that function is defined as seek_length. So now if you want to get the length of any sequence, we can simply call this function seek_length, and then open it with passing any sort of sequence. So here, let's pass it on with this specific sequence. This doesn't have to be this only this sequence, so it's 12. It can be anything. So we can also include like 80, 80, 80, GC, GC, 80, GC. So anything. So this does not actually check for correction or correctness of the DNA sequence. It just considers this DNA sequence as a set of characters. So if there are some inbuilt problems with using our own user-defined way to understand and to, for example, to work with the DNA sequence in any programming environment, because we will not be able to code for all the possible options in which a DNA sequence can be can go wrong, or in which a DNA sequences can have some variations. For example, in this sequence, if I by mistake include some other variety of different characters, our function will not counter for that, or will not take care of that. It does not understand that we are passing a DNA sequence. So hence DNA sequence has certain characteristics, and we have not defined that in this user-defined function. So this is just an example of user-defined function that one can work with. But in order to get, or in order to avail all the different use different characteristics of the DNA strings or DNA sequences, and also use, for example, specific specifically designed functions for this DNA sequence, for working with DNA sequence, we might have to load some few packages and libraries into R. One of the libraries that is extremely useful in defining a DNA sequence in R is a biostrings library. So if you do not have this biostrings library, you might have to install this biostrings library in your computer using BioC manager or BioC manager installed. So that is the what do you call that? BioC manager is the let's check what is that, what that is, how to install that in this one. Install biostrings. Yes, it's bioconductor. I forgot the name of it. Bioconductor has a wide variety of packages that is extremely useful and important for us to work with lot of different biological data. So in this case, it's a DNA strings of DNA sequence. So we can use this specific code that one can get from bioconductor. That is bioconductor. So and then you can Google for bioconductor biostrings install, and then it will come up with this one. So if you run this code, it will install the bioconductor and BioC manager and also install the biostrings package. I have done it. So I will just load the necessary library to do that. So similarly, you can do with the decipher also. I think decipher also is part of bioconductor only. Yes, decipher is also part of bioconductor only, which is also extremely helpful for maintaining and managing biological sequences. So once these two are loaded, I want you to notice the way in which a DNA string is displayed. Until now, our DNA sequence is actually loaded, that is in the sequence 1. If we print sequence 1, so it is going to be printed as a collection of characters, that is A, T, G, and C, or in whatever order that we have. But the moment that we use the biostrings library and use the DNA string function that is defined in the biostrings library to load up a DNA sequence into, let's say, into the object called sequence, and then it is now the whole data class is different. Here sequence 1 is loaded as a simple value, and here the sequence or here the object seq is actually loaded as a DNA string class. So that is the whole object of the object type changes because we are using a specific function that is defined in biostrings library to load up this. Now let's print this sequence. Let's make it a little bit up. Print this sequence and see how it is displayed. So this gives you more than necessary information about our DNA sequence. First, it gives this is a DNA string object. Technically, this is a DNA string object, and the characteristics are defined in DNA string object, defined as DNA string object. And this is a 10-letter code. The length of the code is length of the sequence here is 10, and the sequence actually tells is presented in a color-coded format. So this gives more than necessary information about our sequence. Now we can also use, for example, the length of the sequence option here also to get the length, and it is not actually necessary because we already, when we print the sequence, we can get the 10-letter, get the length as 10-letter. But there will be situations where we have to use the length in inside our code in order to find, for example, let's say if you have 2 sequences and if you want to find out which length is whose length is longer, then we have to use specific function that is going to give the length of the sequence. So here we get a numerical output. This is a collection of strings output which we cannot use it for further numerical manipulation. This is a numerical output which we can use it for further numerical manipulation. For example, let's define another string. So this is seq, and let's define seq1, which is a DNA string, and which contains, let's say, our previously defined longer sequence. So let's take this and then run it. So now I'm actually overwriting the sequence 1 with this object. So the sequence 1 will sequence 1 from the previous with the previous information will vanish, and new information will be stored into the object sequence 1. Okay. Now let's print the characteristic, or generally print the sequence 1. Let's say it's a 27-letter DNA string object with this this one. So again here, if I do the length of sequence 1, and it gives me 27. So I can actually find out which is bigger. Is the sequence bigger, or is sequence seq1 bigger, or is the seq bigger? So we can find out length of seq1. Seq is greater than length of seq1. So it is false. So these sort of options and manipulations cannot be performed using seq versus seq1. Seq1. Yeah. So we get there is an error message because these two sequences cannot be compared in a straightforward comparison. So for that purposes, we specifically need these functions which does specific jobs. So let's move forward and see if you can compute the frequency or compute the occurrence of each and every nucleotide in a sequence. So that we can do using, again, the letter frequency function, and calling the letter frequency function. And this function takes the input data or the input object, and then it takes what to count, right? What should we count? And we say here we should count for, or we should look for the character uppercase C. And then also tells, should I print this out as a probability, right? Or we say, yes, we want to print this out as a probability. Let's take this, and without loading anywhere, and then we can let's see what are the default conditions. Right? At its basic format, the letter frequency should take 2 arguments or 2 inputs or 2 necessary parameters that should be defined. One should define the source data within which we have to count the frequency of what, and the next it should have what should be counted. So let's do this, and it tells, or it gives me just the number of times C occurs in this specific string or in this specific DNA sequence object. So we can do the same for seq1. Again, here 6. So if we use as probability equals true, and then let's see what this one gives you. So this gives us 0.3, which is nothing but 3 divided by 10. So it counts the number of times, or the percentage of occurrence of C in this sequence. Yeah. So let's do the same for sequence 1. So it gives, that is a 22% occurrence of sequence 1. So we can do the same for all the 4 letters, and then print what is the percentage of C, A, T, and G occurring in the sequence. So let's do this one by one. So I'm running the letter frequency option and storing it into object C, and then yeah, option C. Let me pause it for a few minutes, and yeah. So let's use the same code to calculate the percentage of occurrence of all the 4 different nucleotides in our sequence. So the letter frequency function will do that, and I'm going to store the output of the letter frequency into the object C. Then let's print out C separately. Okay, let's see. The lowercase c will store the occurrence or probability of percentage of occurrence of C in the sequence. So we run this, and then we don't get anything. So because we have to print C. So now we get print C 1, and we do the same for G. And then G gets here. And we also have 3 times G in our sequence. Let's verify that. Print seq. Yes, there are 3 G's in our info. So let's count the same for A. Let's run this, and then this gives me. There should be many more. Oh my God! Okay. There should be many more A's. Okay, there is only 1 A, and the rest of them should be T. So people with curious eye would have spotted where I'm actually going with this printing of the occurrence of these letters. So one of the important characteristics that will define a DNA sequence is the GC content, the occurrence of number of times the doublet GC that will occur in any sequence. So this occurrence of GC or GC islands, people would usually would want to call them, is associated with, for example, rigidity. It's associated with higher expression levels. It's associated with many different characteristics of DNA sequence. So it's very important to find out the GC content of any sequence, and that is why one of the quality control methods like FastQC algorithm checks for the percentage of GC in any sequence or in your sequenced reads, that short fragments of reads that I was talking to you about in the introduction part. So let's calculate the occurrence of GC in these frequency of occurrence of GC, the sequence, and then print GC. So we have 0.6 times, but unfortunately, I think there is an error in which G and C are actually calculated here. Let's see. Print C. Yes, exactly. So my reservations were correct. So this actually calculates for G or C instead of G and C. So that is not correct. So that is unfortunately that is not correct. So we have to see how we can include G and C in this specific way. I'm not sure if there is anything that says G and C should be included. Let's say no, it again tells the same of G and C. Okay, vector frequency. Okay, okay. I will clarify this doubt in the next session or after a short break. And yeah, but it is very useful to calculate the occurrence of GC in our sequence. So people who are trying to follow along, please follow along with me until now. You can do it in the R Studio, or you can do it in Colab, R Colab. Also, I actually chose and opted for R Studio because installation of big packages like biostrings and this will actually take a lot of time to complete. So that is why I used this one. Okay, let's continue. There are other ways in which you can actually calculate GC content that does not include letter frequency calculation or letter frequency option, there should be a default option which is or, but we should have an option that is and. Yeah, I am not very sure about the default option or and. Okay, let's see if this one works or not. I found some piece of code which says equals 0. Okay, it says G and C this way. Okay, it gives me this way. So I'm not sure how to calculate the GC content of this one, but maybe there should be some pieces of code which I can explain to you in the next session, in the recording of the next session. Okay, let's move forward. So now we can actually graduate from calculating individual percentage levels of nucleotides in a ready-made sequence that we have loaded up to biostrings or using DNA string option to loading up a FASTA file. So this is the link of the FASTA file that you can get hold of in the associated lesson. For example, let's go to the lessons. So in this lesson, if you scroll up, not this one, so here in this lesson, reading DNA sequences in R, if you scroll down, we have a link to get, we have a link where we can download or where we can get hold of this FASTA file. Or you can use any of the FASTA file that you want to use yourselves. You can actually download the FASTA file to your local computer and then give the location of this specific FASTA file in the R environment. I'm actually using this specific FASTA file link because it is made available or it is made accessible in the over the internet or shared over the internet link, so we can easily load it into any environment. So this is the link which we want to copy the link address. I want to copy the link address for this one and then read this FASTA file as a DNA string. But here if you notice the function that I'm going to use for this is not only DNA string, for which I have used the function DNA string to define any piece of sequence that is loaded in or any piece of sequence that I'm presenting it to this specific function. Here, I'm going to use the read DNA string set function, which will read a file format, which will read a file that is located in your computer or that is located elsewhere in the internet that you can that is made publicly available, and the format has to be in FASTA format. There are also other format you can read in, but FASTA format is a universal format in which sequences are stored, and there are other kinds of information, like if you see here, you can see all the other types of information that you can parameters that you can access, like you can skip number of lines and you can see other information also. So I'm going to use this when I copy that link, this link. This is the link I'm going to copy link address, or you can just copy it, block and copy it. And this is what is I'm going to use here. So let's load up this into the FASTA file, into an object that is called FASTA file. Yes, so it tells you we have downloaded 18 bytes of data and it is being loaded into FASTA file. So if you print FASTA file, then we can, okay, and then we get even more information than printing out only DNA string. When you are going to print only DNA string like seq or seq1, so you got only how long that sequence is, suddenly my R is taking some time to load. Okay, only take how long that sequence is, and yeah, and the sequence itself. So here, yes, and the sequence itself. So here it tells me that this sequence has this specific characters, or the sequence of the object is this one, and the name of the object is something like seq1. It's a default name that is given to the to this object, which we can change it. So we can change the name of this object so that we can call this specific object and use that for further operation. So we can change the name using names function. So you see, this is a value that we are going to change for this specific FASTA file. So we will change that into, or we will load that into sequence name. So that is going to run, and we are going to load that into sequence name. And if you print sequence name, that will be seq1. But the reverse can also happen. So if I say, if I want to change this into something else, let's say my FASTA sequence, and then if you do the same, if you print the name or store the name of FASTA file into this, or you can just print the file, and then it gives the name is changed from seq1 into my FASTA sequence. So you can change the name so that you can better organize your FASTA sequences. So when your FASTA file contains more than one sequence, then each one of these sequences will be given arbitrary name or name in this one. So I think if we open up this specific file, let's say in browser, I just want to confirm if it takes the just want to confirm. Yes, exactly. So the seq1 is the header of the FASTA file. So that is why it is given seq1. So if you want to, if you have multiple FASTA files in your computer, and then that has multiple different headers, your DNA string set will actually read in the headers as their names, and their sequences as this one, so that you can use it for further operations. You can call each and every specific sequence with the name of this, with the name. Like if I want to find out the width of my specific, I think this will work out. FASTA seq, it should actually give you, no, okay, it doesn't work out that way. Yeah, it doesn't work out that way. Let's see how it can work out. File. Seq. Okay, no. Okay, we can find out the width of this FASTA file generally. And then that is going to give you the width of this one. And if you have more than one sequence, I'm going to give you all of them. And each one of them can be assessed with the, from the number as a part of vector or as a part of number. For example, if we have this also there, we can load or we can store the width option into length, and then you can print the length. That is also fine. And now, if you actually want to count the number of different characters or number of different nucleotides in the sequence, then you can use a convoluted function like this to split the sequence into one by one, and then tabulate the occurrences of each and every type of nucleotide. So how do we do that? So this is a function that is necessary to do that. This one will do it. But what are the components of this function? Let's go over this function one by one. This is how we will break it down into workable chunks and then understand what happens here. So let's see what is the sequence actually. What is the, nothing is stored in the sequence. So let's take the my FASTA sequence option and use my FASTA sequence option. So we use the string split function, which takes only this one. So this is the absolute fundamental part of this big or this nested function. So string split function will actually take the input and split it by whatever string or information or splitter that we give here. So I'm going to use, let's see if this will work. Yes, okay. I have to use a specific character sequence for this. So that's why it's not working for seq1, because seq1 is a DNA string or biostrings object. So let's go back to the original definition of defining our sequences as characters like this. Where did I do that? So let's take this and then let's define it here before counting each and every one of them. So here I'm going to define sequence equals this one. Of course, I have to put this into code. So yeah. So if I take this and run, so my sequence is defined as a value that has so many different nucleotides. And then if I use the string split option, str split option for sequence, and if I split it with nothing or with an empty string, so I'm going to get each and every one of these nucleotides are split or actually isolated into separate entities. And splitting this sequence into into separate entities and storing it into a vector will actually enable us to count the number of occurrences of each and every one of these nucleotides. So after this comes this one, right? One. So that is to access only this output in its entirety. So the string split will also give you other outputs, but now we don't want that. So we want to have only the first list output of the string split function. So that is what this specific piece of code does. And then table function will actually tabulate the repeating elements and count the number of times these elements are repeated in your object or in your vector. So let's use table function and then to the output of this string split functions first element. So my R is still trying to find out what I copied. Yes. So if you do that, that is what we want. If you do that, so this is what happens. So it calculates the number of times A occurs in this specific sequence, and C and G and T, all of these, how many times they occur in the sequence. Of course, this kind of information can be extremely useful for us, and hence we can use this information to define and characterize our sequence. Here, let's try. Okay, that does not work out. Yeah, a table would work that way. Okay. Okay, this is how it will work. So you can store this option in, for example, let's say in an object called count, and then use this count option to give out the matrix output of tabulated output of each and every nucleotide in your sequence. So the second associated lesson in the in today's session actually deals with how to calculate the reverse complement of your DNA sequence. So if you click the associated resources, it will have an elaborate introduction to what is a reverse complementary sequence of your DNA sequence and where it is used in the biological uh, 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 relation or biological connotation in the biology of the cell. So I'm not going to go over that. So you can go over that as a part of assignment, and yeah, and understand the biological implications of calculating the reverse complementary sequence of your DNA sequence. So let this load. Yeah. So yeah. So here we start with where these reverse complements are actually usually found and where it is most useful, and in what biological synthesis and what are the associated players in this in the replication, DNA replication, and that uses reverse complements, and it actually tells you what is a reverse complement. Yes, so we all know that from, let's zoom it a little bit, if it can zoom. Yes. So we all know that, as I already mentioned, DNA sequence is always read from 5 prime to 3 prime, and it has a complementary sequence at its other, which is paired and which runs from in the same direction, 3 prime to 5 prime. But we always refer to the DNA sequence in the 5 prime to 3 prime direction. So if the original sequence runs from this 5 prime to 3 prime, and the reverse complement is just a complement that is read from this side, read from the 5 prime to 3 prime for the original sequence. So that is run from here. So for example, the TTT that matches with AAA are actually in the first few, in the first 1, 2, 3 position, because 5 prime is in here. So that is why it is reverse complementary. So it's very important to calculate, or it is very important to it is very easy also to generate the reverse complementary strand for your original strand. So all the necessary codes that is required for you to do this is actually given here. So let's do this in R, and we need the biostrings library again for the same. So the biostrings library has a necessary tool to calculate the reverse complementary of your sequence immediately, or with the use of only one simple function. So when you define any function or when you define any sequence object as a DNA string object like this, let's define this, and then again we know that what this object is. Yeah, it's a 21-letter DNA, 21-letter DNA that has a sequence of this one. And then we can actually calculate the reverse complement of this by simply calling a reverse complement function and passing on our object, or passing on our stored sequence to the reverse complement here. So we can run this line. And then if you print the reverse complement, then that is going to print you the exact reverse complement of this specific sequence. I want you to notice here that since we are mostly using biostrings libraries, it is actually applying all sorts of characteristics, or defining all sorts of characteristics that is associated with the DNA sequence, like coloring the letters, and it will also check for unidentified nucleotide bases or undefined nucleotide bases in your sequence, etc. That will also be reported. If you report something like, let's say how smart this specific library is, if I include something like not you. Yes, so we say the character L is not part of the lookup table, or is not part of the DNA object. So let's say how good this is if I, instead of L, if I give U is part of an RNA string, and that again will not take. Okay, that is good. But you can also use N if you want to use undefined characters in your DNA string. Then N is the one that you want to use. N is the undefined character or a character that we do not know or a base that we do not know what it constitutes. So n is a undefined character that you want to use. So so that will complete or that will conclude the hands-on session for today. So I strongly recommend all of you to follow along as I had demonstrated you how to work with the DNA sequence. And there are some very interesting assignments for you to try it out. First, you should try practicing the code in the or in this try it out for yourself section, where you don't have to type the full code, but you just have to notice places where we have, for example, left some of the sequence, some of the portions incomplete, so that you can try it out. And then once you are successful in trying completing the syntaxes in these two blocks or in these two sections, and then you can go and check for the assignment. Assignment tells you or gives you a space to work with, and then also it provides you a FASTA file which you can download or which you can refer to straight away, and use that FASTA file to create several different or to understand several different characteristics, like the length, like the composition of the nucleotides in your sequence, and calculate the reverse complement of the sequence, etc., etc. So, and there are also some associated assignments in the first lesson, reading the DNA sequence, which should not be that complicated, because it just asks you to calculate the length and calculate different occurrences of AAT and GC using a separate sequence, which we already have tried for today. So yeah, that will conclude today's session. Meet you all in the next session. And thank you very, very much. Okay. So welcome everybody to the second part of today's session in getting started with bioinformatics in Python. We are actually going over working with DNA sequences in Python. So you would have gotten an introduction to what is a DNA sequence and what it needs to, what does it imply in the connotation of cellular function and different cellular cell types, and also what it means to work with DNA sequences from introduction to how DNA sequences are stored in computer. They are stored in the format of a FASTA file, which we can use and load it up in any programming environment like R or on Python, and then work with the sequences to understand the characteristics of this FASTA file in general, or understand characteristics of this DNA sequence in general. So today's session will cover reading DNA sequence using Python, and also how to calculate the reverse complementary sequence that is used in the replication stage of DNA in the cellular context. So the associated resources for this are these two resources. You can open them up and then go through them, and loading data will actually have the necessary information for you to how to load Python, which we saw it last time itself. And this one specific area which I actually left out in describing about Python and how generally to access data from part of data frame, which we discussed last time, how to load a simple table into Python environment, and how to load it as a data frame, and how to access first few columns, and how to access first few rows, and etc. So we did this last time, and I actually left out one portion regarding example Python function, which can be used for many different ways, but generally a function or user-defined function is used to used for those calculations or those specific operations which we intend to do multiple times. For example, if you have to calculate the distance between any 2 points, and then let's say the correlation coefficient of any two data set, then you can use the correlation coefficient function. So, or you can also define the whole set of operations in your own user defined function. For example, that is what we are going to use in Python here. So to work with the DNA sequence, we can simply load up or we can simply store the necessary character into a character-based object or a string object. This is what we saw last time when we discussed several objects for Python coding environment or R coding environment. We can have numerical objects that are vector, which is in 1 dimension, and numerical objects that are in 2 dimension, which are matrices, and objects generally in 3 dimension, which are arrays, which can include all numerical or mixed type or all character type. And when we have an organized table, or when we have a table of multiple different object types organized into exactly same length of rows, then we can use something like a data frame to load up that specific table and then work with that table for further downstream analysis or to understand the patterns of different elements of the specific table or data frame. So, but when we are going to deal with DNA sequences, most of the DNA sequences are from FASTA file are in one direction. Like the FASTA file, let's say, okay, that is the, yes. So the FASTA file sequence is going to be in one direction, where you will have a header information that tells what specific or what are the character, what are the identifier of this sequence, and what organism that this sequence belongs to, and what method can also included in was used to generate this sequence. So that header information is always preceded with a greater than symbol, which symbolizes that what follows is a header, and then what follows is a one-dimensional sequence or collection of characters, that is DNA sequence. So how do we know that's a DNA sequence? Because there are only 4 characters allowed for DNA sequence, A, T, G, and C. So we use the same format to define our own DNA sequence and see what we can do from these sequences in Python. So for the purpose of demonstrating this, I am going to use a Colab or Google Colab in Python, where it is an environment where we can add our code, Python codes, and execute them, and then save it in our Google account. If you have a Google account, it's very easy to get a Google Colab link, which you can definitely get by just looking for or searching for Google Colab in your Drive. Google Colab link, yeah. You can search for this, then you will be given, you will find the Google Collaboratory link, and then you can get a new Google Colab. So this is what is Google. Google Colab.research.google.com is how you will open a Google Colab notebook. So this is a Python environment notebook where you can execute all kinds of Python codes. So in this event, we are going to deal with Python function, how to define a Python function, and how to use the Python function. So a Python function is defined using the character that tells that we are defining a Python function, or we are defining something that is a function. So when we use def, and then what follows is the name of the function, and then what follows after that is the object that this function will actually take in or will actually receive, or parameter that it will receive, and then what follows is a colon, which tells whatever code that follows after this that is intended, right? That is given, or that is that follows a tab. So this is indentation or tabular indentation, whatever code that is intended from this specific colon will belong to this function. So that is how we will define this function. Let's go over definition of this function one by one. So how do we define this? We use the function define, or we use a keyword define, and then seek underscore length. That's the name of the function that we are defining, and then we say it is going to take in an object, and I'm going to name that object as seek. And then I'm saying the definition of the name of the function finishes here, and then when you press enter, it is going to start at an intended position, so which says whatever code follows will be part of this specific function. So this is the function that we are going to define for calculating length of a sequence. So how do we do that? We just have to first define the counter as 0. So first let's define the counter as 0, and then we see while we are going to go over the sequence one by one. So that's how it is. While the object sequence is going to be read in counter one by one, and then we are going to counter plus equals 1. Then we are returning counter here. Counter. So okay, let's see this convoluted statement a little bit and then go over this one by one. It's very simple, and this follows actually what it follows is, let's take this one. So if we had defined counter, okay, I should not have a space here. Yes. So if we had defined a counter as 0, and we are going to define it as 0 here, and let's find out what counter is. Then counter. So it is 0, perfect. So what will this operation do? Let's check that out. Let's find out what this operation actually does. So this operation, counter plus equals 1. What does it do? So we have defined counter as 0, and then we have performed this operation counter plus equals 1. And then when we print counter, what does it do? It actually adds 1. So this is exactly same as equivalent to counter equals counter plus 1. So these two are identical. So to save space and to write easily, so this is abbreviated into plus equals 1. So why is this inside this while loop? So what does this specific while loop does? So there are 3 parts of this while loop. The keyword while, which says while some condition is actually satisfied, do this option again and again and again and again. That's a loop. And this says the name of the loop, or the definition of the first line of the loop ends here. And then the main player is this one. So what does this mean actually? So let's take sequence. It takes counter and then this, and that's all. So what does it do is actually, this is the starting value and a colon, and this is the ending value, and if nothing is given at the step of 1. So that is what this is. So this specifically says whatever is given inside the sequence object, you start at the counter position, this one, and then go by 1. Okay? Increment by 1. That's what it says. So for example, let's say if we are going to define seek equals 80 GC 80 GC. Okay, then okay, let's take a smaller one. So seek equals 80, and then do this, save this, and then let's say seek of 0, with the counter was 0, it actually prints 80 G and C. So that's what it is. So and then seek of 1, it actually prints T, G, and C. So 2, C and C. So it starts with the 0. 0 would mean everything, or 0 from 0 with position, whatever it is, you have to print. That is the notation of this this specific. That's the meaning of this specific notation. And then we increment the starting position one by one in this counter, in this loop, so that we can count the number of characters in the sequence. So you see, when we run this loop manually, starting from 0 and then go to 1, then we are actually eliminating the characters one by one. So thereby we can actually count these sequences one by one. And then once everything is complete, it will come out of the while loop, and then it will return whatever is stored in the counter. That is what this specific function does. So that's a breakdown of this function. Let's define this function. While defining our function, we just have to run it. This is the same. We just have to run it, and it has been it is executed, and that's fine. And here we will execute our function because we want to find out the length of this specific sequence. So this is the DNA sequence that we are going to store into the seq1 object, and we are going to call this function with its name, that is seq length, and pass on the object that for which we want to find the length, which is the intended function of this specific function definition. So we do this, and it gives me 12. So if I add some more, for example, say T, A, G, C, 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 let's say, and then it has to change. So it says 20. So this is how you can actually start with a simple function definition, and then use that function definition to calculate the number of characters or number of letters or number of nucleotides in any given sequence. You notice here one of the difficulty in actually working with the DNA sequence as character is this specific incident. So here I was hoping to type T, but I typed Y, and our function did not care for the presence of any new character in a DNA sequence. So this is what happens when you are also going to work with the DNA sequence, and when you are going to use not any specific libraries or any specific packages for working with DNA sequence. So we are not going to use any packages for working with DNA sequence in Python today, and there are 2 or 3 reasons for that. One of the main important reason is to install a package in Google Colab. It takes a long time, and and managing that will be difficult. But if you want to work with DNA sequences in Python, so you can check something like BioPython. BioPython has some excellent libraries for working with DNA sequence, and there are also other libraries that you can use for working with DNA sequence in general. So we will proceed further, and for our next operation, we wanted to actually count the number of sequences or number of bases in each and every number of A's, number of T's, number of G's, number of C's in our sequence in a FASTA file, not in any simple characters that we are going to enter. So for that, we have to first read the FASTA file into load the FASTA file and store the contents of the FASTA file in an object. So for that, we can actually use another function or define another function, user defined function, which we will take and use it to read the FASTA file. That is for which we are going to perform some operations on. So the function that we are going to use is actually defined in this lesson. Not in this lesson, in the second lesson, for example, in the associated resources, you will have read DNA sequence using Python. So that specific lesson will have the necessary function. So by now, I think you would have gotten used to what a function can do and why we need to define function. So here we also have some introduction to DNA, which you can read. And for reading in Python code, DNA in Python code, we have some, let it load. It's taking a while. Yeah, let's let it load. So this is straight, this function is straight from this lesson. That's what I wanted to tell you. Yes, here, this is where I find this specific function. This is where this specific function is there. It's we can take it straight from this library, this specific lesson, and then let's see. So for this function, we need to import a library. We import a package called collections. Right, from collections, we want to import counter. So counter will actually count the number of occurrences of different bases or number of occurrences of different letters in the in any text or in any object. So for this, we want to define a function that will open up a FASTA file from your local computer and then read the contents of the FASTA file. So if we actually look at the contents of the FASTA file, as I mentioned, the FASTA file has two portions or two parts. One part is the header, and the second part is the sequence of the FASTA file. So for this specific usage, we are going to use something like an object that is that's a specialty of Python environment. And that is the object called dictionary. So just like in any physical dictionary, what you will see is you will find a keyword, and for that keyword, you will find all possible explanation or the meaning for that specific keyword. You will search for that keyword in the alphabetized arrangement. So similarly, dictionary is just like any physical shelf, and you can place at different boxes or different positions in the shelf, different things in the shelf. So something like that. Python's dictionary has an index or a key and a value. So this could be the container, or this could be the shelf, and this could be the contents inside the shelf in your physical analogy. So this can contain anything. It does not have to. It does not have to be specifically numeric, and it does not have to follow everything has to be numeric. It can contain a mixed number of objects, types of objects for the values here 10, 20, and also for the keys, right? So that's what it is. This is how a dictionary is going to be called. So if we use this dictionary, it's going to be called by calling the key or calling the index of that specific dictionary, and then that will give out this this value of that specific dictionary. So it's like a named array. So for array and data frame and everything, we'll have numerical positions that will define which row and which column that this specific value is from. For dictionary, the position or the address of that specific value is defined by us. So we are going to use this this powerful object type of Python, and then going to incorporate into reading our FASTA sequence. How are we going to do that? It's basically simple. We are going to define in the FASTA file, we are going to define the index of the file as to be the header, and the value of that specific index to be the sequence. So that is what we want to do. So that is what it is. So that would mean the NC 001542.1 rabies virus, the complete genome, this becomes the container, or this becomes the index, or this becomes a key for this specific sequence, and the sequence value is going to be this. So let's go back to the Colab and see how we are going to implement this. So we are going to simply implement this by first importing the counter from collections package. So we will do that. And then we will start with defining the function, and we start with defining the function, open and parse FASTA. Always use meaningful definition of the function or meaningful definition for the object, which is going to be extremely important for us. So it is going to take file path. So file path is nothing but the path of the file or location of the file that we are going to refer with. that we are going to store it in an object called file path and pass it on to this specific function, where this function is going to use the file path for opening the file. So it is going to open the file using open file option. So using open function, and that says f of what is that file path. This says, wait a second, okay, there are three components of this one. One component is which says what to open. I want to open a file, so that is mentioned by the character f. And where is that file located? And that file is located in this specific location, or this specific file path. And what do I want to do with that file? I want to read with that file. That's what these components are. So then as I mentioned, we are going to load this FASTA file and load it as a dictionary object. So dictionary object is actually used by defining something on the object. So we are going to define an empty dictionary object first. FASTA underscore dict equals this, and then we are going to also define an empty label, or the header information label equals column. Okay. Notice that the moment that I define the function, the next lines are coming as intended, or belonging, or enclosed within that specific function definition. So this is how Python works. There are no brackets, or there are no flowery brackets that defines the function or defines a loop in Python. So the flowery brackets are restricted for use of the other uses. So here I'm going to start with a for loop which says for line in FASTA file. One of the advantages of actually using the open function is it will take the take whatever the contents of that specific file, and it can read line by line as defined as line, right? For line in FASTA file, what are we going to do? For each and every line in FASTA file, what are we going to do? Because FASTA files can have multiple lines for the same sequence, and these lines can end at 60 or 100 depending upon the width of the width setting. But until and unless the FASTA file encounters another header character or nothing, or a blank line, everything that follows belongs to that one specific sequence defined by that header. So that is why we are using the line option to parse or to go over multiple lines of the FASTA file, just in case if our FASTA file has more than one line. So what is this going to do? And we are reading this FASTA file line by line, and in that line we are going to use line.r strip, as it wants to remove, or we want to remove the new line spaces, or we want to remove any space, or we want to remove any new line characters in the FASTA file. There could be some space that is that comes before the start of the first few characters in the FASTA file, or there could be a new line, specific new line character that tells go to the next line. And these characters are hidden characters that you cannot see generally, but Python's compiler can actually read them, so we have to get rid of them. So we are left only with the DNA sequence. So we have to use this R strip option to remove spaces and new line characters for each line. And then comes the meaty part. So now we have excluded all the unwanted characters, unwanted white spaces, and new line characters from our FASTA file. Now we want to figure out whether the line that we are actually considering in the FASTA file is a header line or the sequence line. So the only main difference between these two parts of the FASTA file is that the header line starts with a greater than symbol. So we are going to use that option to find out whether or not the line that we are actually considering is a header or not. So we use if condition which says that specific line has a characteristic that starts with the character greater than symbol or not. So if it is the case, then we are going to name that line as the into, or we are going to store that line into FASTA underscore label object. So that is going to be this specific line 1 is to whatever. So this we already saw what it does. We already saw what it does in the counter operation on the sequence operation. Why the line starts with 1? It's, if you had guessed it until now, then it's very good, because if the line starts with 0, then our FASTA label will start with the identifier which is greater than symbol. We want to get rid of that identifier or greater than symbol from our FASTA label, or from the label which is not wanted at this case. So our line array, or our not array, our line vector actually starts with the position 1. That is why it starts with 1 and ends at until the end of the line, and we are storing that line into FASTA label. And then we say, we also, okay, not here, and after this we are also going to use FASTA dictionary underscore dictionary. Remember, we defined FASTA underscore dictionary here, and in this FASTA underscore dictionary we are going to define the key, or define this the holder which will hold the sequence. Now we have identified the header line, so that is what we are going to use it as a holder. So FASTA or key or an index, whatever you want to call, equals nothing. So we have not encountered anything here, so we say it's an empty until, I mean, right now it is empty, and we are going to define the holder here, which is, or the key here, which is a FASTA label, or which is the header information. And then next we are done with whatever has to be done within the if condition. We have to go a little bit back, so I type backspace, then else again, if the line doesn't start with a greater than symbol, it has to mean that the line is part of the sequence. So if the line is part of the sequence, then the line has to go inside the FASTA dictionary, which defines a sequence under the key, or under the value, under the index, the header index. So we have to define that one here, FASTA underscore dict FASTA label equals so here plus equals line. So the necessary operations for these is also defined there. So what it actually does is, it actually takes the line and then puts it next, or appends it to the FASTA dictionary. So the line here should actually, when it satisfies this condition, contains only the sequence information of the FASTA file, not the header information. So we are appending or adding the sequence one after the other, one after the other, one after the other, in just in case if our FASTA file contains multiple lines. So we are done with defining this function, and then we have to send back whatever that we have stored so far to the main menu, or to the main to the call which calls this function. So that is done using return function. And what do we want to return? We want to return the whole FASTA dictionary. So this is how we are going, this is what we are going to use for counting, or we are going to use for this specific function. Okay, let's define this, and it worked perfectly without any problem. So let's see if this one works or not. How do we see that? For this, in the associated lesson, we have given the file. Where is the file? Here. You can download this file to your local computer and then put it up into your Python environment, refer to the Python environment. If you are going to use a collab like this, what you have to do is, you have to look here, you have to click on this folder option, and in this folder option there is a way to upload to session storage, and from this upload you have to upload that specific FASTA file into here. I have already done it, and then if you upload it, it will be available here. and from here you can right click, copy path. So you do this, you copy path, and then you use that path to take this. So what do we do that with that path? We will use that path to submit or to give it to open and parse function that we have just loaded, right? Or just we have just we have just defined open and parse FASTA. I forgot FASTA, so FASTA. Okay. So we can actually do that and load the contents or load whatever is actually done or whatever actually executed from this specific function into the object. Let's say FASTA sequence. Into that, let's do that. And it is executed. Let's open it up and then print it out or display it. And yes, that is going to display in this specific format, which is how a dictionary is going to be displayed. A dictionary always is displayed with a flower bracket as given here, flower bracket with the index, with the index followed by the value. So similarly here, a flower bracket with the index, that is the header of this specific FASTA file, followed by the FASTA file as such. So this is a huge FASTA file, and then we already have actually defined a count function. We can use this count function to find the sequence length of this FASTA file. Let's try that. To do that, we have to use seek. I think what is the name of that function that we have defined? Seek length. I hope this worked. And then inside seek length, we have to give FASTA underscore sequence, and then it's NC. Yes, this is what it is. Yeah, this is what we want to give. And then let's run this. Let's see if it works or not. Yes, it definitely worked. So this is how we will call the content of the dictionary. So we have to mention the value, mention the key, or mention the index of the dictionary to call the content of the dictionary, and that is done by calling calling the a header, which is stored as an index in our dictionary. That's how we have defined the FASTA file in the function. So our file contains something like 159,662 sequences. That's excellent. So many values. That's very good. So this is what we did, and then now within this sequence we can actually define another for example a function to count each and every base, right? There are like 160,000 bases. So here nucleotides, here we can actually count how many times A occurs, how many times T occurs, G occurs, and C occurs separately. So that is what this specific counter this does. So let's see if we can use this counter straight away instead of using it in a function, because we can actually do that. Let's say if it works that way. What happened here? Yes, because you remember from collections package we imported counter. So this is what we are using, FASTA underscore seek, then NC. It's not working. Okay, we can copy it and paste. Maybe it won't work here. I don't know. Some functions are actually conducive to use within with the dictionary one. Some are not. Let's run this. Let's see. Yeah, it does. Fantastic. That's what we need. We don't even have to define another function for this. So we can open it separately and then load it into FASTA underscore sequence and use the use the dictionary to call it, or we can put an intermediate step which actually loads the sequence of this one, sequence long of this one into this, right? We can actually put it that way. Let's see if this one works. I will just print this. Let's see if this one works or not as an independent entity. I think it works. Yeah, that's why it's taking a long time to run. Yes, so this one works. So this should actually work. So it's better to do it this way. So I'm going to save the first, say the sequence that we have stored in the dictionary into seq long object, and then I'm going to count the number of times A, T, G, and C appears in this specific seq long object. So counter should actually give me, yes, it does. So let's take this also and then put it somewhere. So I don't want to anything. So this we did after this. I cannot open up a code. Yes. So here we can use the same sequence length function that we have defined before, and then we can use the seq long here, so that we can we don't have to keep mentioning this, which is long and makes the readability really poor. So this is what the counter collect one does, which is which gives an extremely important statistic. I think we also can do the same counter for more manageable sequence, right? This is what we defined here before coming into here. Seq1 we have defined as a seq1, so we will define it as seq1 again, and then let's see. In seq1 it's very simple. There are 4 A's, C's, T's, and there's a Y. That's amazing. That's good. Because I mistyped seq1 yeah, T for Y, so that's also getting counted as a part of counter operation. So this will be extremely important if you want to calculate how different types of nucleotides constitute our DNA sequence. So in the lessons, we have actually given a function to do that. I think you can check that out, and yeah, you can check that out, and I think you can follow the definition of that function, as I have already demonstrated you how different functions are actually used, how functions are used in Python's classes. So similarly, another associated lesson will actually deal with calculating the reverse complementary sequence of your DNA sequence. So yeah, that is going to in order to understand where reverse complementary sequences come into play in the cell's functionality, we have given an introduction to DNA replication and what are the different components and different enzymes of the DNA replication process, and how this is actually making use of the reverse complementary strand, and etc. etc. So reverse complementary is nothing but, if you take the sequence at its usual direction, in which we are defining a DNA sequence, that is 5' to 3' end, and the complementary strand of that specific strand, or that specific DNA sequence, a DNA strand, in the same direction as the original strand, goes from 3' to 5' end. But our usual conventional way of defining starting and ending of a DNA strand is from 5' to 3' end. So in order to give a complementary sequence for any given sequence, the complementary sequence also has to be displayed or has to be presented only from 5' to 3'. So automatically the complementary sequence becomes the reverse complementary of the original sequence. So this is what we have to carry, we have to calculate. So for that to calculate, we can actually find out what is each and every character of the original sequence and what will be the complementary to that specific character or to that specific nucleotide, and that can be calculated. And once the complementary is calculated, this can be reversed from the end to 1 in this order, so that what we get out is a reverse complementary sequence of the original sequence. So let's go to Python and see how this is implemented. And this is implemented until now we have finished it. This is implemented again as a part of function. So let's go over this function one by one. Once this one starts responding. Yeah. So this is there are two ways to define this, functions to define this. One of the function is, again, we start with defining a function by def keyword, and then we give the name of the function, reverse complement DNA. And then what does it take? It obviously takes a DNA sequence, and we give that DNA that as the object with which this function is going to work with. And now we have to define a complementary dictionary or complementary dictionary. So that is what we define here. We define the name of the dictionary is complementary, and then that is defined as a dictionary which takes a value would mean T. See, that's how it is defined here. Dictionaries, yes, here. So we are defining a dictionary here again as a concept of dictionary. A would mean T, as A's complement is T. So dictionary can be used in several ways. And similarly, T's complement is A. And then we are defining the rules here. And then G's complement is C. And then C's complement is G. So once we have defined it, and then we can actually use a convoluted statement like this, which takes return not okay. I will explain you one by one. Join reversed. I think yeah, it is little bit easy to it's readable. I for I in DNA sequence. Okay, let's break this down. So what happens here is, why does it say I'm missing sequence? Yes. So this specifically says for I in DNA sequence, give what complementary to I is. So that is what is going to be given. So for if you take this, we have not defined a DNA sequence again, and we have not defined complementary. So let's define complementary first, and then so let's define complementary. So complementary dictionary is defined, and now let's break this code down. This can be written as something like this: for I in DNA or seq1, yes, 1. What does it do? It give me, please give me print complementary of I. So that is what it's going to give. Okay, print I also, I, and then hopefully this one works. Yeah, complementary of I. And then let's see if this loop works or not. Yes, it does work, and with some y error, because we have a y there and there is no y in this one. So let's, yeah, for just for the moment, let's ignore about the error, but it gives the complementary strand A. I mean, we have printed I also, so it gives for whatever sequences. We can actually print the seq1, so that we have a reference here. Yes. So for A, it gives T, for T, it gives A, etc., etc. That is working. So that is what this actually does. This one big chunk is what actually does that. That's what it does. So for each and every letter or nucleotide in DNA sequence object, You give the complementary of that specific nucleotide. But if you do that, let's see if I do take this and do that as this one. So we already know what this one does. So let's don't use this one for it. So instead of DNA sequence, let's put it in seq1. Let's also name or rename seq1 so that it doesn't contain the Y character. Yeah, and then let's see what this one does. Yes, it is going to give you complementary in this way. So it's going to give you in complementary for this way, only the complementary inputs for this way. And then we have to reverse this. So how do we reverse this? We just use a function called reverse. Reversed is going to give you, and I'm going to copy this reverse. Let's see. This is the normal one, and then let's see, reverse is going to give you the absolute reversed for this one. Something bad happened. Okay, that's okay. Okay, it will work with this one. I think it will work with this one. So that's why it is not working here. That's okay. Reversed off, maybe that one will work here. No, attribute error, okay. Okay, this should work. I don't know why it is not work. Reversed. Okay, reverse iterator, that's fine. Okay, okay, just reverse the iterator and we have to join it. And how do we have to join it? When we are going to join, we have to specify what are we using to join. So we are here actually using nothing to join. So join it with the reverse iterator. It's just a reverse iterator, that's why it's not working. So if you do this, it's going to give you in the reverse order, but it's going to join without anything. This empty symbol actually tells you what joins these two characters. If I include comma here, that's going to add a comma between each and every character, each and every nucleotide. But that's not how we want our sequences to be. Yes, see that? So that's not how we want sequences to be. That's why we have a empty character there and we want to join, and the reverse did not give any output because reverse just gives the index of this array. That's what it does. So it takes the index of this array and then reverses the characters, and then it just returns that specific output to wherever it is being called. So that is what we actually did. Where is our defined definition of the function? I actually lost track of this. Yeah, okay, I think it's here. So let's define this, and then we have defined it, and then let's find out what is the reverse complement DNA of seq1. So if we do that, it's going to give you exactly the same answer. Can you do it for the longer sequence? Yes, of course you can do it for the longer sequence, but that is going to take long time. So we don't want to use the longer sequence. But what we can do is we can use the longer sequence, but not from everywhere, from 0 to maybe 100, maybe only until then we can use, yes, maybe only until then we can use it. So we can reverse complement of longer sequence, but only from 0 to 100 nucleotides. And that is going to, given that is okay. Reverse complement is not defined because I have to define it as reverse complement underscore DNA. Yes. Yeah. So we got this. So this is a very good way of understanding functions, dictionaries, and a lot of powerful options from Python, and also at the same time working with DNA sequences. So we understood how FASTA files can be stored into a Python environment, how we can find length of the FASTA file, how we can find different, how we can count different nucleotides of that specific sequence, and how we can reverse complement them, and etc. etc. There is another way to reverse complement the DNA sequence, that is using nested replace option. So that is what is actually used in the Python lesson, and that is, this is nothing but simply it takes the DNA sequence and then replaces each and every sequence with the corresponding letter. It replaces A with T, and then after it is done, the whole sequence, it replaces T with A, and after it is done, in the whole sequence, it replaces C with G, and after it is done, it replaces G with C, and etc. Here, there is an excellent use of the case sensitivity of Python in place. You see, you observe this. For example, in this multiple sequence of replaces, you see all the complementary nucleotides are actually lower cases. And what is the need to include them as lower cases? Because the moment when I use uppercase here, A will get replaced with uppercase. All the A's in the sequence will get replaced with uppercase T, and immediately after that, again the T's will get replaced with uppercase. So there is, there will be a difficulty, or there will be a mismatch between, or our reverse complementary sequence will not agree with our original sequence. So what this piece of code actually elegantly does is, it takes the DNA sequence, or takes the object, and then uses the replace attribute or replace option to replace all uppercase A with all uppercase T, and then after that is done, in the second part, it replaces all uppercase T with all lowercase a, and then after that is done, it takes all uppercase C with all lowercase g, and then after this is done, it replaces all uppercase G with all lowercase c's. And then once all of this replacement is done, so what we have now is not reverse complement, but from forward complement or complementary strand of our original sequence in lower cases. So we have to change that into uppercase. That is what this function does. And after that, what this function does is, this is just an abbreviation for, this is just an abbreviation for start, stop, step. So this is like reversed. Reverse will give you the index, but this will actually reverse the sequence. So when nothing is mentioned, it will start at 0. When nothing is mentioned, it will stop at the last point. And when the step is mentioned in the negative, it goes from back to forward. So that is what it does. Nothing is mentioned here, it goes from the start, and nothing is mentioned at the stop, it goes to all the way to the length of the sequence, and then -1 is mentioned, so it goes in the reverse direction. So we first replace all the uppercase A with lowercase t, and then we replaced all the uppercase T with lowercase a, and then we replaced all the uppercase C with lowercase g, and then we replaced all uppercase G with lowercase c, and now we have gotten the complementary strand, not in the reverse direction but forward direction, all in lower cases. We then changed that to uppercase, and then reversed it with this specific shortcut. So then we store all of these into a reverse complement object, and that's how this function works. And this is the function that is given in the lesson. And let's run this function and see whether this function actually works for the same piece of code, or works for the seq1. If I can get hold of code, yes. So now our reverse complementary DNA is actually using this method. Maybe I should change it. Okay, we should change it to DNA1. Let's change it so that that is DNA and this is DNA1, and we'll use that also. So reverse complement DNA. What happened here? Reverse. This is spelling wrong. Okay. Yeah. Okay. Yeah. Here, complement DNA of seq1 is this. This is using the dictionary method. There is no choice. I mean, there is no one one method better than the other one. We can use anything you want. I just wanted to tell you two elegant ways of using dictionary and multiple nested replacements to come up to the same conclusion. Yes. So that's how we can perform Python operations on DNA sequences to get reverse complementary strand and other calculations. So this will conclude today's session, and I urge you all to go through the associated resources, because there are some extremely interesting assignments and them, and we give you practice yourself area where you can practice all the code that's been explained today here, and also all the code that's been explained, that is explained in the reading in the lesson itself. So you can do that, and in the lesson 4 in the reverse complementary, I think we have a we also have a practice running code. We give you a different file for which you will have to calculate, or for which you'll have to display, find out the reverse complementary sequence. This is not the file that I showed you, but this is a different file. So I insist all of you should go and complete these two assignments, practice these two cases to get a better understanding, or to get a better understanding of how you are following Python lessons for accessing and working with DNA sequences. Thank you very much for, thank you very much. That will conclude today's session. Let's meet in the next session, where we are going to perform a lot of exploratory analysis of the structured table that we were discussing in the last session. Thank you very much.